All right, let's bring on our next speaker here. Myron Holubiak is the president and CEO at Citius Pharmaceuticals. If the chat, if social media chatter is any indication, we seem to have a lot of people that are very interested in this company. So, Myron, good afternoon. Good to see you.
Good to see you, Spencer's. Good catching up with you too.
Yeah, good catching up. All right, let's go ahead and share your screen. I see it up there. There we go. I'm going to turn the floor over to you. Tell us about Citius.
Thanks, Spence. Thanks very much. I'll put up the standard disclaimer statement, but there's, as you notice, I've highlighted some things. So please, if anybody's interested in investing in Citius, we strongly encourage you to view our company's filings. And just this morning, we filed our 10Q and also an 8K with a press release that was filed today. So a lot of the material that I'll be discussing, some of you may not have been familiar with because we've updated a lot of information. And the company itself, the name comes from Citius Altius Fortius, which is the Olympic motto, and of course ours means faster. And the reason we chose that name and kept the name even after we merged, it has to do with the speed with which we develop products. And the strategic decision that we made to pursue 505(b)(2) products initially. So that's where the company started. We've added to that strategy. We now are looking at a lot of the interesting opportunities. We currently have 4 active programs, and our lead asset is in finalizing phase 3. And you can see there's a little description there, but I'll get into more details shortly. All the markets are very large. And the products are unique. So we had a strategy of 505(b)(2), which are usually substances that have been in man before. But one of our criteria was that it had to be completely unique. So it had to be a first and only. And in this case, we have a market. We call it CRBSI, stands for catheter-related bloodstream infections. That market is well over $2 billion worldwide today. I'll get into more detail shortly. The ARDS market, whether it's associated with COVID-19 or not, is fairly large. It's 250,000 incidents here in the United States, over 500,000 around the world with no real therapy available. Then there's tissue expander, TEs, tissue expander infection. And the great story here is that post-mastectomy, women who decide to have breast implants later frequently use a tissue expander in that intervening time period, and the infection rate there is very high, even with current standard of care antibiotics. So we've decided to tackle that as well. And then finally, when we merged with Citius, they had a prescription product being developed for a hemorrhoid market, which initially we didn't think we'd be interested in, but once we learned more about the prevalence of hemorrhoids, particularly grade 2 and 3, and the fact that there are no prescription products that have been approved by the FDA, we decided this is a very important opportunity. We have a great team here. I'll show you their history. We've just added to that team. And our advisors really are the opinion leaders, whether we're talking about the bloodstream infection, infectious disease market, or pulmonology with ARDS, and also breast surgery with our tissue expander product, infection prevention product. We're extremely well funded. That's part of what we filed this morning, has brought everybody up to date on our finances. And both my partner and I, Leonard Mazer, who many of you may know, have invested over $26 million in the company ourselves. So you know that we believe in what we're doing. These are, to me, I'm going to focus on the 7 and 10 pin. These are the executives of the company, with Leonard and me being the original founders, Jamie being our CFO, as well as others. But Dr. Chutchman, and here's a Dr. Myron Chutchman that it's the only company you'll ever see with 2 Myrons in the executive group. And Ilana Allen, who recently joined us, are 2 that I'm going to focus on a little bit. Chutchman is our chief medical officer. He's exec VP. He recently came to us as from being vice president of clinical research and development in lymphoma at Celgene. And prior to that, he was at the Roswell Park Comprehensive Center, and he was chief of the lymphoma service. He's published a lot of articles, and he's taken over as chief medical officer, as well as helping us direct our efforts in the areas that we're intending to develop. Ilana joined us very recently. She's got a lot of experience in investor relations and corporate communications. She's helped us even with this presentation that you're looking at right now. She has an excellent background and has accomplished a lot in her career. These are our active programs. You see Mino-Lok there. It treats central venous line infections. Again, I'll get into detail shortly. It is in phase 3. It's actually a very late phase 3, and we're ready to do another drug monitoring review. Again, I'll get into detail shortly. Halo-Lido is our prescription therapy for hemorrhoids. We're in a PIND phase. We're ready to file the IND. We're awaiting the FDA's response, which should come any day now, in terms of some specificity around the endpoints, as well as the run-in period to do the trial. But we feel it's all in very good shape, and we'll have an IND shortly, and we'll start our phase 2B trial this year. Mino-Wrap is the product I was alluding to relative to preventing the infections associated with tissue expanders post mastectomy. We've had a PIND discussion with the FDA. We probably will file our IND early next year. I think we've tried to get that in this year, but we've had to respond to the FDA communications as to what the model needs to be. And then finally, more recently, in last September, we got involved with a gene development company, gene editing company, Novellus, and we formed the sub, which we're calling Novocyte. And we've already started a lot of work on our what we call induced mesenchymal stem cells, and I'll spend a little bit of time on there. That IND should be filed early next year. Minlock is our lead product. You can see a picture of it. It's 2 vials. One vial contains minocycline. The other vial has a combination of disodium EDTA dissolved in alcohol. And this is an antibiotic lock therapy. It's for salvaging catheters that cause bloodstream infections, particularly in vulnerable populations. The problem with central venous lines, particularly in cancer patients and hemodialysis patients, is they frequently become infected. And when they do, they're very life-threatening. So we have about 7 million central lines implanted every year in the United States, and up to around 450 to 500,000 can lead to serious bacteremias. And they're associated with 12 to 25% mortality and morbidity. It costs a lot of money to deal with the CLABSI. I'm using CLABSI and CurbSI interchangeably. They're slightly different, but for our purposes, we can consider them to be the same. But the DRGs and bundled payment system is somewhere in the $50,000 range, and of that, about $10,000 is used for removing and replacing the catheter. Those are 2 separate procedures. And that's where our product comes in. So Minlock would be the first and the only therapy under investigation that could be used to sterilize and salvage an infected catheter, avoiding all the complications, the tremendous discomfort and costs of removing and replacing an infected catheter. So that's what this program's all about. A little background on the R&R complications is there's infectious complications. They occur 26% of patients. Mechanical. The catheter breaks, the tip is misplaced or something like that. That's 5 to 19%. And thrombotic, so you get a blood clot 2 to 26% of the time. And below you see all the sources for this information. But on this slide, I want to draw your attention to this physical and psychological symptom burden that also is presented by CLABSIs. Well over a half to 2/3 of patients do report that they have significant physical and psychological symptoms associated with catheter removal and reinsertion. And that was a study that we did alongside with MD Anderson. MD Anderson actually did the study. We helped fund it. And that is something that's new information that people are not yet really aware of. With us, it's quite different. Instead of removing and replacing, you basically leave the catheter in, and what you do is you sterilize the catheter. So you fill it up with our solution. The solution sits for 2 hours. So for 2 hours, it's dwelling in the catheter. It is locked. It does not drip into the human body. At the end of those 2 hours, it's aspirated and discarded. You flush the line with saline, and you reinstitute the venous line. This is done for 5 to 7 days. So 2 hours, 5 to 7 days. And to date, we've been 100% successful in salvaging the catheter. This is our phase 2B trial results. And this is a 90-patient study. The control arm was remove and replace. So a brand new catheter was introduced. That therefore microbiologic eradication, as you can see in this line, is the same. But you also notice there's a quicker relapse in the new catheter. But I draw your attention here, though, more importantly, the complication rate is in the 18% range. And there's a variety of things that happen here. And these could be severe adverse events. There's different kinds of complications that occur. These patients are relatively the same relative to the bacteremia experience. Our patients actually in our arm were a little bit sicker. We had more ICU admission and more mechanical ventilators than the control arm did. But nevertheless, we were very impressed with that. We took this to the FDA. They also were impressed. And they've authorized our phase 3 study. However, they did ask us to compare it to standard of care antibiotic locks, which is what this trial is. And this is the trial that we're doing right now. What we're looking for right now is the difference between, we call it time to catheter failure. It's basically a standard metric. And we're looking at 21 versus 38 days in a 42-day week period. 6 weeks is the test of cure. We think we're doing great. We're well along in this particular trial, and we're ready to go for our superior efficacy analysis that'll happen this quarter. Here is our timeline, and actually I'm going to update this from the date that I have on here. But you can see here we sit in the second quarter, and our drug monitoring committee meeting will happen in the second quarter. At that time, we expect to get one of two answers or directions. One of them being stop the trial, you've proven your point, which we have our fingers crossed on that. The second one is everything's going fine. Again, this is efficacy as well as safety, and we're trying to prove superiority. So this is a superiority trial. And in that case, we may have to take this further to the end, but we're well over 80% of the patients right now. So we expect some good news happening this quarter. If not, we'll have to extend it a little bit. But we do expect that this year the trial will be completed. The market opportunity is huge. While, as I mentioned earlier, it was somewhere in the 450 to 500,000 prevalence rate here in the United States. Worldwide, it's much greater. The infection rate is measured, the metric is how many infections per 1,000 catheter days. We brought that down here in the United States to 1.2 per 1,000. The rest of the world, particularly China and India, are somewhere in the 6 to 8 per 1,000. So you see that this is dramatically needed in the rest of the world. And we think that we finally have a solution that really hasn't worked very well. Antibiotic locks, which I alluded to as our control in the study, don't work that well. And the reason is that the organisms form a biofilm, and that biofilm is very resistant to antibiotics. Our intellectual property takes us to 2036. It's held by University of Texas, MD Anderson Cancer Center, where we license the product worldwide. And there's a series of patents that protect the IP. Regulatory protection, we have qualified infectious disease product. We have fast track designation. We also get some supplemental protection in the EU. So this product has at least 16 years, but certainly 10 years of protection. The only other product that's being developed here is Cormedix, which we expect will be soon launched. But Cormedix is very different. We are for treatment. You use us when the catheter is infected and the patient has bacteremia. Cormedix is used when there is no infection, and it would be used in hemodialysis patients only thus far today. So the summary of Mino-Lok, which again is our lead product, it treats catheter-related bloodstream infections. It penetrates biofilm. It eradicates the bacteria. It salvages the catheter. And people are highly dependent on these lines, so it's a very important thing. We expect to be indicated as adjunctive therapy for the treatment of CRBSI, and we have worldwide rights for a long time. Another area that we're working in is with the mesenchymal stem cells. As I alluded to, Citius has partnered with Novellus, which is a gene engineering company in Cambridge. We formed a sub, and that sub is working on developing mesenchymal stem cells for the treatment of acute respiratory distress. Here's why we fell in love with this and the compelling advantages that our cells have. We use a synthetic messenger RNA cell reprogramming process to create our pluripotent stem cells. Most other people are using donor-derived cells that have their own issues relative to supply and also potency. So we have a clonal iPSC master cell bank. It's a consistent cell source. So there's no batch-to-batch variation. It's footprint-free. There are no viral vectors. We don't use that. Again, we're using a synthetic mRNA process. It's fairly rapid production. It takes 4 weeks for us to differentiate the pluripotent stem cell into a mesenchymal stem cell. We call them IMSC because these are induced from pluripotent stem cells, not donor-derived. We have greater expansion ability. Ours can expand 70-time population doublings. Compared to something in the 10, 12, 15 area for donor-derived cells. We've shown that we have higher levels of secretion of many immunomodulatory proteins compared to donor-derived. And they will be allogeneic, so they'll be off the shelf. They'll be supplied in frozen, ready-to-use mini bags. We're using it in ARDS, primarily associated with coronavirus right now. I think you're all familiar with ARDS. It basically is very destructive. It causes damage to the epithelium of the lung, and mesenchymal stem cells have shown some promise here. This is why we think we have an advantage. Our IMSCs, again, the synthetic derived iPSC-derived cells compared to bone marrow-derived cells, have huge advantages when it comes to the secretome itself. Again, this is in vitro data. But we've done a sheep study, and this is the result of that. We have very strong beneficial effect using our IMSCs on sheep models that have acute lung injury where we've induced ARDS. So this is very indicative that we have something exceptional on our hands. So the description will be allogeneic, iPSC-derived induced mesenchymal stem cells, very high potency, footprint-free. There's no viruses used. They're clonal cells. They're telomere restored, meaning they have more population. We believe they're superior MSCs. They'll be available to treat acute inflammatory respiratory conditions. Here's the timetable on that. Right now, we still have to do some work. We're involved in our preclinical studies right now and some of the in vitro work. We expect the IND to be filed early next year.
I'm about to do something that will make me very unpopular, but I'm going to jump in here, Myron. We're up against the clock.
Okay.
There was a lot of questions. I think he actually answered most of them. There was a lot of questions about timelines, and if you weren't paying attention or if you missed it, just rewind the stream back 10 minutes. There was a great slide up on timelines, so check that out if you missed it. We got a lot of questions about the offering this morning. Those via selling shareholders. I don't know if you want to speak to that very briefly as far as what you'll be using that for, but after that, we got to wrap up.
Okay, fine. Let me jump to that. And we did have a fairly good raise this year, and we now have roughly $104 million in the bank. That's enough to take care of all the programs that I've just described to you. The offering, there was no offering. What we basically did was register some warrants that needed to be registered. We're not planning on doing any more offerings in the near term, unless we have an opportunity that requires it. We do have to increase our common stock as well as some stock options. We're starting to hire a few people, as was described. So this is the cap table as what was reported this morning. I think we're in the roughly $300 million market cap range, and we're trading at just above $2. So, why should you invest? We have unmet medical needs. We got a very attractive, diversified portfolio. Our research partnership is primarily with MD Anderson Cancer Center and 2 of the programs that we're doing, and with Novellis on the stem cells. Lots of milestones are anticipating. We're very near the end on our major product. A lot of seasoned executives with a very successful track record. So that's what Citius is. We hope you consider it. We don't have any plans to do any raises in the very near future.
Myron Holubiak is the president and CEO of Citius Pharmaceuticals. A lot of people are here just to hear you speak. Myron, thanks so much. It was a pleasure to catch up, and just to give everyone some backstory. Myron, I went to kindergarten with Myron's son, so we go way, way back. And I'm not saying it makes me an expert in Citius, but I'm not saying it doesn't either. It's my point. All right, Myron, thanks a lot.
Thanks very much. Thank you.
Those of you that are here just to hear Myron speak, do me a quick favor and hit that like button. Subscribe to Benzinga. We're doing a giveaway when we're going to hit our 100,000 subscriber on YouTube sometime in the next week or so. And when we do that, we'll be giving away some free stock and some other expensive things. So subscribe to Benzinga on YouTube. Hit the like button.